Good morning or afternoon or whatever it is, wherever you are. It's a pleasure to be here to talk to you. I'm Ted Kalbfleisch. I'm an associate professor in the Department of Veterinary Science at the University of Kentucky. Welcome from the state of Kentucky. Okay, so what am I going to talk to you all about today? Translational science is something that's been pretty popular in human medicine for I guess the last 15 or so years. What is it? Let me see where I can, there we go. Historically, there have been three domains of research. There's basic research that is essentially the testing of hypotheses with in domains that aren't immediately applicable to the practice of medicine. That's chemistry, that's physics, that's engineering, that's molecular biology, all of those sciences. So clinical research is another domain where after years of testing a particular chemical for a particular function, somebody gets the idea to turn it into a drug, and will recruit patients and will test the efficacy of this particular medication. And then there's clinical practice, where you have physicians seeing patients, identifying diseases, prescribing treatments for those individuals. And in each domain, the clinicians and the researchers who are doing the work are writing, they're giving presentations just like we are today. But most commonly what happens is their findings get written up and published to the peer review literature. The entire process of information exchange between these domains historically has relied on individuals from, say, clinical research reading a paper by a basic researcher, getting the idea that there is a drug that can be developed based on information that has been derived in basic research, turning that into a drug or into another treatment, testing that, publishing that, and then ultimately it finding its way, if it is effective, into clinical practice. So this has worked, but couldn't it be better? So what if we were in a position where instead of doing our work and simply throwing it over the fence into the peer review literature, which is a most honorable way of distributing our findings, disseminating our findings, what if we were actually talking to one another? What if we were exchanging ideas, and identifying people across these domains who had a shared common interest with us? So what the term for the way that information and knowledge is aggregated and sort of held is has become silos, right? So our research is siloed. We build all of this knowledge, but really it doesn't get out freely or easily as we wish it would. So how can we break these silos? Again, the notion is we communicate, right? We build actual human bridges between these domains of research and practice, and identify people with whom we can collaborate. The NIH has they back in 2006 they implemented what is called the Clinical and Translational Science Program. And what they identified was that the best environment to drive the translational research paradigm, they were medical research institutes. Because typically they were homes to these three domains. And if Oftentimes these folks were working in buildings that were either right across campus or even right across the street from one another. If you could get them together talking and exchanging ideas, well, things are just going to get better. There we go. So again, the objective, getting scientists and clinicians with common interest to communicate. Okay. What translational science is not, and let me be clear about this. What we don't want to do is turn clinical practitioners into basic researchers, or God forbid, try to turn basic researchers into clinical practitioners, right? These are very different domains that require very different skill sets and knowledge sets. But what we would like to be able to do is get clinical practitioners, clinical researchers, and clinical and basic researchers essentially thinking like one another, or at least thinking in ways that would allow them to communicate more effectively with the people who are working in these other research domains. Okay, so bioinformatics is an enabling technology. How can this happen? Or how can this even help? I'm going to ask that question for you. Data volumes are very large, but they're structured and they're queryable. Wow, structured and queryable data. You're going to tell me, I say I'm a bioinformaticist, you're going to tell me that there's structured queryable data out there. I'm going to go get it. So we have begun collecting data. So back in the day, maybe there are a few people here on the call who are old enough to remember when you looked in an article in the literature, data. Well, so what was data? Well, back then, right, it would have been a text table in a publication that would likely had 20 columns and maybe 6 rows. Or strike that, reverse it. There was sufficiently small data that a graduate student or postdoc or even a professor themselves could write those data down in their research notebooks and begin to use it. The data volumes that we are working with right now are prohibitively large. They are impossible to work with in that manner. So the entire the entire recorded or everything that has ever been recorded in human history in terms of literature and journalism and so forth would fit on about a terabyte of data. We're generating data for individuals at a rate of 100 gigabytes a clip. Yeah, so moving stuff around by paper, by emails, by PDF files, it's just not going to work, right? What you need is to store these data in organized fashions where it can be queried, and that's what bioinformaticists do. High throughput genetics data has created an extraordinary opportunity in this regard, and I'll talk a little bit more about that later. Okay, well, I guess I'll start to talk about it now. What has been the most fundamentally enabling technology in translational research has been the electronic medical record. Again, those of us of a certain age can remember walking into a doctor's office and we would see rows and rows and rows of shelves with racks and racks of files, right? These were paper copies of medical records that were faithfully recorded and stored, and their privacy maintained by simply keeping people away from the files. Okay, this isn't going to fly anymore. And it was recognized back in the early 2000s. What we need to do is we need to move to a domain where we are storing these data electronically in databases. Vital statistic, diagnoses, prescriptions, radiology, all of these things can be stored in ways that can be readily queried, that can be easily shared, and that can be analyzed in a relatively straightforward fashion. Other fun things that are getting stored are pathology samples, archived blood, things of that nature. Things that if we wanted to go back and test them for some other hypothesis or for some other piece of information, The samples are there for us to do it, and typically they are linked to medical records in the EMR. And then genetic data by way of SNP chips and now even high throughput sequence data are being generated either as a result of direct to consumer interest. So I have a 23andMe SNP chip. I also have an Ancestry.com SNP chip. That data is out there for me. I have access to it. And heck, why wouldn't I want to make that available to my physician? Okay, bioinformatics. So in a nutshell, bioinformatics is where molecular biology meets data science. So if we as bioinformaticians can access the electronic medical records, this gives us phenotype data. So because the thing that we need is accurate phenotype data, access to samples, and access potentially to genetic data and SNP chips. We can do statistical analyses. We can access genome annotation, and we can see, by virtue of genome-wide association studies, what regions of the genomes are implicated in certain disease or pharmaceutical reactions. Right? This is incredibly powerful. We could begin to understand the nature of inherited disease or disease susceptibility. Right. So what happened in the early days of clinical and translational research was, let me go back a bit. What happened in the early days of clinical and translational research were scientists zeroed in on this. They said, okay, if we get these data in these medical records, that's very powerful. But what would make it vastly more powerful is if we also stored the samples and the blood. If we can do this, then we can do genetic data. We can run SNP chips. We can even do a whole genome sequencing. We can pull the samples. We can do that rapidly. We can do virtually any genomic analysis that we want to. Okay, so genetic data of animals. Well, so a brief history lesson here about high throughput sequencing. The Human Genome Project was wrapped up, I don't know, around 2002. Cost $2.3 billion and took about 10 years. And in terms of manpower, all the king's horses and all the king's men. Hundreds of scientists worked on this project. A lot of money, a lot of time. I can generate the same amount of data that was generated for the whole genome sequence project. If I had a machine, I could do that in a couple of days for a couple of $100. It is really that dramatic. The word unprecedented is thrown around a lot these days, a lot of times inappropriately. This is a very appropriate use of that word. $2.3 billion to $600. Yeah, if you want to drive the price of anything through the floor, give it to the biotech industry. Right now, there are high density SNP chips for cattle, sheep, horses, dogs, cats. This is data that can be used for, and really these are the most simple uses of these data. Parentage verification, copy number variation. And a copy number, for those of you who don't know the field terribly well, is, there are certain regions in the genome that simply that are functional, that get themselves replicated or otherwise removed from the genome, imparting different phenotypes to the individuals who have those genetic signatures. We can do association studies, that is, know what regions of a genome are inherited in common by individuals who share a particular phenotype. Then there's high throughput sequencing. So high density SNP chips cost between $100 and $200 a piece. And, this is a vastly underutilized data set. Lots of pet owners are generating these data just for the fun of it. Just to get back some clever report that was generated. In the hands of a genetic researcher coupled with solid phenotype data, these data are incredibly powerful. Also, these data are being used in cattle, sheep, pigs for genome-based genome-based breeding programs or genome-assisted breeding programs in order to improve production phenotypes. So SNP chips, again, between $100 and $200 a piece per animal, which is something that consumers are readily willing to pay for their animals, and even livestock producers are willing to absorb those costs for their breeding stock. High throughput sequencing, however, whoa, this blew the doors off of everything. So it's species agnostic, it's disease agnostic. Essentially, what you do is you take a sample from an animal, you prepare a library, which is a fancy word for essentially you break up the genome, you put adapters on it, you slather it over a chip, and then you sequence it. And whatever is in the tube, right? So the sample from your animal, whatever bacteria was hitching a ride, if the laboratory technician sneezes, whatever is in that tube gets sequenced, and we get to analyze it. It's incredibly powerful. So whether you're sequencing, I don't know, a gerbil or a chicken or an alpaca, you would prepare the data. You would prepare the samples exactly the same way to go on the machine. You would analyze the data only slightly differently in that you would map it to a reference quality genome to put it in the context that is best to understand it. So the chicken, you sequence the chicken, you'd map it to the chicken genome. You sequence an alpaca, you'd map it to the alpaca genome, etc. But once it's there, right, you can look for variation that might plausibly cause a specific disease if you know the genes implicated in that disease. These are the data sets that are being generated at 100 gigabytes per animal per clip. Okay, so opportunities for translational science and veterinary science and medicine. We essentially, so in human medicine, we're patients, we've got doctors, if we do 23andMe, there are pharmaceutical companies out there that are kind of interested in making medicines that are going to make it easier and better for us to live. Our data, when we go to the doctor, is stored in these electronic medical records. Essentially, that same ecosystem exists in the veterinary world as well, right? You have pet owners, you have veterinarians with electronic medical record systems, you have animal pharmaceutical companies, and you have these direct-to-consumer providers who are generating genetic data for animals, as well as breed associations that are generating these data for their elite animals. Okay, so genetic, simply restatement of that point. These technologies have been transformative in human medical research and in animal research. What do I mean by that? That's a bold statement. All of science prior to about 1995 was aimed at testing hypotheses. So you developed a hypothesis and you tested it. These data made it possible for us to generate new observations that allowed us to generate new hypotheses. And the mode of operation was effectively known as hypothesis generating research. Right, so we really had no idea. We had some idea of how these molecular biological pathways were interacting, and what gene produced, what transcript produced, what protein that interacted with another protein produced by another transcript in another gene. So we had some idea of what these things were, but we had no idea how they worked in concert with one another. These chips gave, and the sequencing technology gave us the ability to probe these things in ways that we never had before, and gave us the ability to look at things differently and ask newer, much more exciting questions. Okay, metagenomics. Remember I said anything in the tube gets sequenced? Well, somebody recognized not too terribly long ago that we were very much minorities in our own bodies. So, the bacteria in our gut, and in our airways, and things that are working productively with us as commensals, we can know what a healthy metabolome looks like. We can know, for a healthy person, what bacteria is supposed to be there, and how it is working with us to help us. So sequencing stool samples gives us a very clear idea of our gut microbiome. Sequencing throat swabs gives us a very, or nasal lavages gives us a very good idea of the bacteria that exist in those systems. So these are things that we can sequence and we can understand. And when we have cataloged everything that's supposed to be there, when we see a sick animal, we can do the same. We can do the same sort of sample collection and very quickly find out what their bacterial populations look like, and how they may or may not deviate from that which we see in a healthy animal. Okay, remote data generation. I think this is incredibly exciting. So there's a company named MatMacorp. They build this instrument that is capable of running 8 PCR reactions simultaneously. And this is something that can plug into the 12-volt adapter in your car or truck, right? Or they even developed some sort of clever solar array that you could plug this thing into if you were in a remote part of the jungle. But if you are on a farm and you have a sick animal and you've got to dose this thing with an antibiotic, you want to know what's the bacteria that I'm dealing with, so that I can know what the right bacteria or what the right antibiotic is. So you swab the animal, and in under 2 hours, you can have a result that gives you a better idea of exactly what is going on with this animal. This device, which is simply incredible, this is a sequencing machine, right? So again, for those of us of a certain age, sequencing machines were nearly always these things that were roughly the size of a refrigerator, and they made a lot of noise, and they needed specialized cooling and computer equipment and this and that. Well, this thing is about the size of, I don't know, what used to be called the cigarette lighter, and it plugs right into a computer. Right? Everything that you need remotely to generate a lot of sequence data, to study whatever, right? You want to, and it's completely unbiased. Again, you're out in a field somewhere, and you want to know what bad things might be swimming around in this particular watering source. You can sample it right there. You can test it. You can tell the farmer what's going on their farm. And again, I think this is incredibly powerful, and it's all been made possible by bioinformatic analyses. So obstacles, goodness knows there are obstacles out there. And some of them are that, yeah, I've said that there's a lot of data, but it's sparse, right? So, if you went to somebody and said, okay, give me all of the data that's out there, you would be hopelessly overwhelmed. If you went to a clinical office and said, give me all of the information that you have on this patient, well, you're not going to get, almost certainly you're not going to have everything that you need. And what we need, what I would very much like to see us do, is get ourselves to a position where we start collecting routinely the phenotype information, the vital statistic information that would be necessary to perform these hypothesis generating experiments downstream should we need to. Of course, there's data security and ensuring authorized access. That is hugely important. I'm not, to be clear, I don't want to gloss over this at all, but it is its own domain of expertise. And those those are people that need to be engaged with any of this, and none of it is possible to be done responsibly without them. And then another thing that's going on is, of course, the notion of integrating systems and their data in a distributed computing environment. So everybody's going to have little bits of this and that here and there, making certain that all of these, the electronic medical records, the genomic data repositories, and the software that analyze these things are all what we call interoperable, meaning that they store data in common formats, that the query languages and that you would use and the queries that you would use within them are all relatively common, and would make it possible to ask exciting questions across all these domains. Okay, so I will conclude here by saying there is a lot of information-rich data out there in veterinary medicine and in veterinary research. And bioinformatics is a field of endeavor that can enable a more rapid exchange of ideas and dramatically speed up hypothesis generation. And I mean, what we want to do, right? A physician sees a patient and recognizes a disease and doesn't have any good idea based on existing scientific findings what they should do next, right? That is why the entire research domain exists, to get to the bottom of that question, right? So if that physician would know how to pick up a phone and call somebody that they could begin to discuss the problem and to discuss how to get to the bottom of it, then that physician is going to be that much closer to being able to treat the next patient that comes in with that problem. So rapid communication of ideas, the culturing of common problems and common solutions for them is what all of this is about. And bioinformatics, I would continue to suggest is a key to making all of that happen. So acknowledgements. I work for the University of Kentucky Department of Veterinary Science in the Gluck Equine Research Center, and they are ever so kind as to provide me with a paycheck every month and make it possible for me to do fun stuff like this. So it has been a pleasure talking with all of you, and I am more than happy to entertain any questions.
Thank you for that valuable talk, Doctor. Now the stage is open for questions.
I have a quick one.
Yes, sir.
For the remote sequencing device you showed, which is very interesting, how much depth do you get in terms of doing things genome wide? How does it compare?
Yeah, I believe that device can produce 1x or 2x something of a large mammal's genome. So you're not going to generate a mammal's a mammalian genome with it, but you can, if you're going after bacteria, bacteria would be child's play for that. So that said, there's our new technologies, new approaches out there where we're building haplotype maps that allow you to impute whole genome genotypes for an animal. Say, an Angus, you got an Angus, impute whole genome genotypes for that animal with only one-fold coverage. I don't know that the accuracy is going to be good enough on that device to be able to get you there, but those technologies are out there, making it possible for you to conceivably get whole genome genotypes at 1x.
That's very, very interesting, and definitely could be a major advance of putting it in the field. So that's very nice.
Indeed.
Thanks.
Indeed. Yes, sir.
Thank you for answering the questions, Doctor.
My pleasure.
Once again, I thank you for being a part of this webinar and hope you associate with our future events in person. Thank you once again and have a great day, sir.